Assalamualaikum and greetings. My name is Nur Fatina binti Manisa, a final year student of Bachelor of Science Honour in Biology, and today I'm going to present my final year project with the title Bisphenol A and Alzheimer disease, a review of clinical evidence under the supervision of Mr. Razif Dasiman. Dementia is currently the 7th most common cause of death in the world and one of the illnesses with the highest societal cost. According to a report by the World Health Organization (WHO), there are 35.6 million people worldwide who suffer from dementia, which is expected to more than double by 2030 and quadruple by 2050. Alzheimer disease, the most basic form of dementia, disrupts message-carrying transmitter in the brain, particularly those in charge of memory storage, by destroying brain cells and nerves. Alzheimer disease is characterized by widespread atrophy of the cortex and deposition of amyloid plaques and neurofibrillary tangles of hyperphosphorylated tau protein in neurons, which contribute to their degeneration. According to the National Institute of Environmental Health Sciences, Bisphenol A (BPA) is widely used chemical in the production of polycarbonate, a hard, clear plastic found in many consumer products. In a 2014 report, the United States Food and Drug Administration (FDA) stated that exposure levels of less than 5 micrograms per kilogram of body weight per day are safe. However, the new research suggests that even at the safe level of BPA exposure stated by the FDA, it may contribute to a variety of health problems. Numerous research has proven that BPA significantly increases oxidative stress, neuroinflammation, amyloid-beta, and tau-phosphorylation in the Alzheimer disease brain tissue cortex and hippocampus. Therefore, this study aims to advance knowledge of the potential involvement of BPA in the development of this neurodegenerative disorder by summarising the experimental research on the connection between BPA and Alzheimer disease. The objectives of this research are to determine the health consequences of BPA consumption with clinical evidence, to identify the harmful impacts of BPA on the biological system that lead to the cognitive impairment, and to provide an insight into the relationship between BPA exposure and Alzheimer disease. There are 3 research questions for my review article. First, what are the health consequences of BPA consumption? Second, what are the harmful impacts of BPA exposure on the biological system that lead to the cognitive impairment? Third, how does BPA contribute to the Alzheimer disease? There are several research gaps in the existing research. Although many studies have examined the adverse effects of BPA, there is a lack of research with clinical evidence on how BPA triggers the development of Alzheimer disease. Besides that, the unexplored molecular mechanism that mediate BPA-induced brain damage and its impact on Alzheimer disease are still not well proven. Also, the need for a better understanding of the effects of BPA on cognitive function, neuroinflammation, and amyloid-beta build up. Bisphenol A or BPA is an industrial chemical that is commonly used in the manufacture of plastics and is added to a wide range of consumer goods. For example, polycarbonate plastic bottles, beverage and food cans, dental sealant, and thermal paper. BPA is a synthetic substance that belongs to the diphenylmethyl derivatives and has 2 hydroxyphenyl groups linked to the carbon atom of the main chain. It is a colorless solid that is soluble in most common organic solvents but has very poor solubility in water. BPA is classified as an endocrine disrupting chemical (EDC) due to its ability to bind endogenous hormone receptors and cause serious effects. BPA has been investigated by public health agencies in many countries as well as by the World Health Organization. There are many adverse effects of BPA that have been proven in clinical studies. It promotes cancer, reduces fertility, induces neurological disorders, heart disease, promotes obesity, and diabetes. While normal exposure is below the level currently associated with risk, several jurisdictions have taken steps to reduce exposure on a precaution basis, and particularly by introducing BPA-free plastic, which are manufactured using alternatives bisphenol such as bisphenol S and bisphenol F. But there is also controversy around whether these options are actually safer. In a person with Alzheimer disease, toxic changes in the brain destroy the healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves 2 proteins called beta-amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates and eventually forming tangles inside neurons. And beta-amyloid clumps into plaques which slowly build up between neurons. As level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Achieving a deeper understanding of the molecular and cellular mechanisms and how they may interact is vital to the development of effective therapies. Numerous research has been made in identifying various underlying factors to Alzheimer's disease. First factor is insulin pathway disruption. Yu-Ming Wang, 2022, indicated that BPA exposure impaired cognitive function, disrupted the hippocampal regular cell arrangement, increased blood glucose levels, and disturbed the signaling of insulin pathway, including phosphorylated insulin receptor substrate 1, protein kinase B, and glycogen synthase kinase 3 beta. The second factor is neuroinflammation. Yin Hua Ni, 2021, found that the impacts of BPA on cognitive function exhibit a site-specific manner, such as altered diversity and composition of the gut microbiota, decreased production of SCFA, damage to the intestinal barrier function, and the BBB increased the neuroinflammation and colonic inflammation, inhibited the 5-HT metabolic pathway and reduced 5-HT levels in the hippocampus, serum, and colon. The third factor is vascular complications. Ending and Dog, 2021, showed that BPA significantly inhibit the transport function of BCRP. Additionally, BPA was predicted to bind to the cavity that is targeted by known BCRP inhibitors. Taken together, the findings demonstrate that BPA inhibit BCRP function in vitro, probably the direct interaction with the transporter. This effect might contribute to the BPA known impact of neurodevelopment. In conclusion, based on the analysis made in the study, there is a potential link between bisphenol A, a widely used industrial chemical, and the progression of Alzheimer's disease that has been proven by clinical evidence. The current study has focused largely on clinical and experimental data on the neuropathology of Alzheimer's disease and its potential with the BPA. However, the study also acknowledged the need for further research to better understand the molecular mechanism underpinning the BPA-mediated brain damage and its effect on neurodegenerative disorders. The study concludes that more research is necessary for conclusive results and to develop effective prevention and treatment strategies for Alzheimer's disease. These are the list of references that used for this study. That's all from me. Thank you.